So good afternoon, tēnā koutou katoa. Thank you very much to the organisers for this opportunity to give a talk. I'd like to just start off by acknowledging the people that I've had collaborations and with over the years, and the number of students that I've had who have worked with me, and noting the support of the Humboldt Foundation, the Alfred Sloan Foundation, and what is my home department. So I thought I'd like to say something reasonably philosophically challenging today, if I possibly could, and see whether or not I could change people's minds or get them to think in a different way about biology and the role of genetic information in biology. So I'll just start off with a bit of an introduction, then get to talk about replication and translation, and then I'll give my characterization, my different characterization, I think, of molecular biology, and try and back it up with a discussion of the mechanism of quasi-species bifurcation. Well, you'll find out what I mean by that when we get there, and then I'll come to a conclusion. So I'm going to start here. Before he drank the hemlock, what did Socrates have to say? Well, he was a bit disillusioned with science. When I was a young man, Seabees, I was an enthusiastic devotee of the branch of wisdom known as natural science. I thought it a glorious achievement to know the causes of everything, why each thing comes into being, why it continues in being, and why it perishes. I was always worrying myself over such questions as these. Does heat, by undergoing some sort of fermentation, give rise to animal life, as some people say? Is blood the instrument of our thinking, or is it the brain alone that supplies the sensations of hearing, sight, and smell? I once heard someone, it was Anaxagoras, reading from a book. The author talked about such absurdities as air, ether, and water, as if in trying to state the causes of a particular action on my part, were to say, he is now sitting on that couch for the following reasons. His body is composed of bones and sinews. The bones are hard and jointed, while the sinews are capable of contraction and relaxation, and together with the flesh and skin that contains them all are laid around the bones. And therefore, as the bones are slung loose in the ligaments, the sinews, by relaxing and contracting, enabled him to bend his limbs, and that is the cause of his sitting there with his legs bent. So it's a bit of a critique of mechanical causation. And he says this is utterly absurd to describe bones and such like as causes. True enough, I couldn't have carried out what I consider to be right, that is, except the death sentence, unless I had bones, sinews, and the rest. But to say that these things are the cause of my doing what I do, and that I act with intelligence but not from any choice of what is best, would be an extremely slipshod way of thinking. So I'm going to apply this to biology and say, what use is it to try and understand biology in terms of the laws of physics and chemistry? Okay, so the first thing we notice about biology is we've got something computational at its center, and that's the genetic code, which relates the base triplets to the amino acids as they're going to appear in order in the sequences of proteins. I would like to use the word computation in the way that Zdenka used it this morning, and suggest that maybe we could exchange that with information processing, the term. And then if we look at it from a physical perspective rather than a computational perspective, the way in which the genetic code is actually operates in organisms is in protein synthesis at the ribosome. And so you have this messenger RNA here. It's divided into codons of 3 bases, and for each one you have a tRNA with an amino acid attached that must come and line up in the appropriate place, and then the amino acid is transferred to the growing chain. But how does the correct amino acid get onto the transfer RNA, which is going to correspond to the bases? That's the question that we have to address if we're going to actually think about the genetic code, because it's when those amino acids are attached to the transfer RNAs that the code actually operates. The rest of it's just a stringing together operation. So those enzymes that do that are at the center of the story that I'm going to be telling. So I'm going to have a rather extravagant thesis here. I'm going to suggest that the conjunction between physical processes and computation, the code and ribosomal protein synthesis, represented by the emergence of the genetic code, I'm going to suggest that this rewrote the laws of the cosmos, at least locally. Look out the window, all those trees, everything virtually on the surface of the earth is made up of little tiny cells, and they are packed full of little computing devices, if you like, those ribosomes, which are making proteins. Nowhere else in the universe, unless there's a similar form of life, is there that density, that organization of matter that is computationally directed and controlled. You don't see that anywhere else. Start from the Big Bang, go through the transitions of the different forces separating from one another, and then it could just keep going. Everything that physics tells you, you would never get to biology from that unless you thought about the manner in which computation can intervene in matter to make that happen. And when it does intervene, you get a completely different sort of matter, organized in a completely different fashion from anything else in the universe, from stars, from planets to interstellar dust. Nothing is anything like biological material, especially from the point of view of its degree of organization and its lower entropy anyway. So my second statement there is that the inviolable laws of physics and their inexorable consequences were subjugated to the control through constraints, what you would want to call guidance. And that rendered the laws of physics causally impotent, practically irrelevant except as a potential nuisance. And that's what the laws of physics amount to in biology. You have to work out how they apply and what happens, but they don't tell you anything about biology in the end. They're just a bit of a nuisance. So people have tried to answer that question or look at it in various ways, starting with Schrödinger in his famous book, What is Life? And he said that the important thing about living systems is that they contained a code script for their construction in the form of an aperiodic crystal. That's what he called it. And that's very like the structure of the heteropolymer DNA or RNA or whatever. The other thing they had to do, that they have to do continually in order to be alive, is they have to export the entropy they produce, if we can call it export, because there are irreversible processes happening all the time. That's what metabolism is. And you wouldn't be alive if you weren't doing that. You may be frozen in a freezer somewhere and you can slow that down and stop it. And you do that with some frogs and thaw them out and they jump away. But as long as they are, that order can be preserved in that fashion at low temperature, but once it gets going again, there are irreversible processes happening. Then Jerry Joyce came along and he tried to persuade NASA of what they should be looking for when they go out into space. And he said that life was a self-sustaining system capable of Darwinian evolution. Didn't get us very far either. I think we get much further, much closer with the statement which Paul Davies and Bernd Kuipers have used at various times, saying that life is matter plus information. But that doesn't satisfy me either, because information is something static, it's just there. And unless it's processed and unless it's interpreted, then it doesn't mean anything. And the whole point of biological systems is that you have genetic information and then around it you have a cell capable of interpreting it. If you had the genome of the human being out in space, you'd never know what a human being is, and you couldn't deduce what it was without first of all knowing what a human being was, or at least knowing how biology had evolved on Earth. So the information in itself is nothing, it's meaningless. It only acquires meaning in conjunction with the interpreter, that is with a human cell around it. So I'm going to try and describe life as reflexive computation instantiated in molecular processes. And when I'm talking about computation, I'm just going to be talking about the manipulation or transformation of information. But I'm going to look at all computation in the way that Zdenka mentioned to us this morning, that it must be instantiated in some hardware domain. It's got to be physical information. It's got to be actually doing something, not just some abstract idea that Turing has in his head and develops into a formal system. I'm not looking at computation as a formal system, but as something like the way in which a automobile factory is a big computational device, it's a robotic one. It's real world computation that is being done. And information, I'm just going to take to be patterns of whatever it is that you choose. You can take, you can measure the Shannon information content of anything if you know the probability of the individual things that you're talking about happening, and what the probability of each outcome is. And I'm going to label the outcomes, and that's what I call an alphabet, the letters of an alphabet. So now let's get down to the way in which we see molecular processes actually happening in biology. The thing which is most peculiar about biology is this thing that I call reflexivity. That is something in which you've got production that requires the product, the chicken and the egg problem, which came first. And replication is the simplest version of this happening, where you take something, I'm just calling it A, could be an organism or whatever, you take components and you can make a copy of that organism as bacteria do when they reproduce. And so you get 2A out of 1A plus a whole lot of components. You can draw that in 3 different ways. Here I've just said, well, here the components get transformed into A, but it needs A to be there. And this is where you see the idea of reflexivity. Components get transferred to A using A to conduct the transformation. So this sort of replication was studied in detail by Manfred Eigen in his book, sorry, not his book, his paper, Another Wissenschaft, in 1971, and he called the paper Self-Organization of Matter and the Evolution of Biological Macromolecules. And the 2 main results of that paper really were the demonstration in such systems of Darwinian evolution, and what you see there on the left-hand side is just the survival of the fittest. The species with the highest reproduction rate increasing until it eventually dominates, and the other 3 species, or only 4 competing here, the other 3 eventually dying out. But this can only happen when the replication has a certain low enough error rate. So that's in the diagram on the right-hand side. Did I say right before I meant left? And as the error rate increases from 0, the final equilibrium, as we call it, selection equilibrium of this process, if you have perfect replication, you'll end up with species X4, a proportion of 1 in the population. And then as the error rate gets a bit higher, you'll come to places where actually there's more of the 1 error mutants than there are of what you would call the master species here. And if you take a slice through there, a slice anywhere through there, then you get the population distribution of each of the species, which the individual species which are present at equilibrium. And the population which you have present at equilibrium, you call the quasi-species. It's a mixture of the master molecule and all its satellite mutants around it. Now, if you increase the error rate above a certain threshold, then that whole selection process disappears. And you just stay at this point here where everything is present at in equal proportions as the starting point as you were there. So that was the main result of Eigen with respect to replication as an example of reflexivity. Another example of reflexivity that you have is an autocatalysis. And Stu Kaufman was really the person who set about preaching, I suppose, about autocatalysis. And it's been taken up by Mike and Wim Hoordijk and others. And the point of reflexivity in autocatalysis is mostly to do with catalytic closure, that the components that act as catalysts for the reactions need to make all of the components in the system. And so you've got this reflexive relationship between what is made and what is there. And that's what defines an autocatalytic set. And here's an example. You can overcome the chicken egg problem in autocatalytic sets by having food molecules which are available and connect as the sources for everything. And one of the characteristics of autocatalytic systems of that sort, networks like that, is the idea of critical connectivity. As the system gets more and more connected, you reach a threshold value here, after which it's very easy to form autocatalytic sets. But when the connectivity is very low, you're less likely to do so. Okay, so how does all of this relate to self-organization? Well, in the case of Schrödinger's idea, it's energy dissipation which is required for the maintenance of order in the system. And that's very close to the idea of Prigogine and his dissipative structures, and there being dynamic instabilities between different regimes in the dynamic phase space. And in far from equilibrium systems, you're likely to have that occurring, especially when you've got feedback and nonlinearities in the ODEs describing the system. And a good example of the kind of order that you can produce is the Turing type of reaction diffusion systems, which produce spatial patterns. And then, of course, there are also temporal patterns, chemical oscillations that you get. And of course, the Belousov-Zhabotinsky reaction is the one which has been taken as the classic. And this form of molecular self-organization occurs within the molecular thermodynamic phase space. So it's a concept that really is derived from physics, but it's anticipatory of biology in the sense that if you look at the way biological systems are composed, they are far from equilibrium, they are driven, and they have a lot of interactions between different processes, so they're highly nonlinear and they're likely to become ordered. So this was a way of thinking that Prigogine talked about while he was alive. It was the innate tendency of driven systems that degrade energy, like a feeding organism on the Earth's surface fed by sunlight, the intrinsic tendency of those to self-organize and reach states of order, lower entropy, which would not be predicted from the simple laws of equilibrium thermodynamics. And just to pay attention to the Zhabotinsky-Belousov reaction, this is a time series that Prigogine published in his 1971 paper, going from left to right and from top to bottom and left to right. This is a reaction starting off in homogeneous solution, eventually producing a state in which you've got the spatial separation of different chemicals in different concentrations up and down the test tube. Happens absolutely spontaneously. You have to have good temperature control to see that clearly, but of course it doesn't last forever. Once the food has been used up, the food of the reaction, it'll disappear due to diffusion. But as long as you've got the reaction diffusion coupling occurring and maintained, you can get those stationary waves in something as simple as a test tube from a chemical reaction. I mean, if you're told from all the teaching that you do at secondary school about the second law of thermodynamics related to chemical reactions, this just blows the mind of students when they see it, because as Zhabotinsky said, Belousov tried to publish his results in peer-reviewed journals, but eventually gave up after referees and editors insisted that such behavior contradicted the second law of thermodynamics. And Zhabotinsky took it up and was a champion of it. Okay, so now I want to move on from there to autocatalysis as it applies to genetic coding. And once again, I've got a picture here. Wish I could get this mouse to work better. Here we go. Of a messenger RNA, ribosomes attached to it producing proteins. That is done by taking tRNAs and amino acids and joining them together with these enzymes, the amino acyl tRNA synthetases. You attach the amino acid to the tRNA, and that is used to make the proteins. But note that you need proteins for the ribosome, and you also need proteins because that's what the enzymes are. They're proteins. And so you've got the sort of closed causal loop there. And if you are going to have the system operating, you need reflexive information. So the idea of reflexivity applies to information as well. That is information which, when translated according to the rules of the code, the proteins produced operate the code. You must have reflexive information to operate a code. And so we can represent the coding self-organization in this fashion, that you have amino acids, which are being turned into, amino acids are getting turned into proteins, but we are choosing the amino acyl tRNA synthetases here, the coding enzymes, they fold into their normal form, but they are required in order to make themselves. So coding is in fact an autocatalytic process. The entire set of the assignment catalysts, that is the coding enzymes, is typically required to make any one of them, because most enzymes of any length are likely to contain all 20 of the amino acids. So this is the idea of reflexive information. Now, there are a number of us, Colombano starting in 1977, then Bedian in 82, and then I took it up in 1993, not knowing that they had done it beforehand, and did simulations of a system like this. And so I took a 4-letter alphabet, and so I started off with a message gene there, and then the probability of any particular amino acid being joined on corresponding to a codon was chosen at random to start off with. There was no coding at all. There was just the ability to join amino acids together to form proteins. And so the proteins that were formed were absolutely random. Then eventually, as a matter of chance, there were a group of proteins present in the solution which had some activity for a coding set, making unique assignments of codons to amino acids. And when you get them there coincidentally, they then translate that information into something which has slightly stronger activity for those particular coding assignments. And then you get a very, very rapid transition from random to coded assignment, and eventually all of your populations, because that's what the gene with the reflexive information contains messages for, all the proteins are then coding enzyme proteins, and the whole thing has reached the highest level of activity that it can. So this form of self-organization in the population of proteins is possible only because you've got the reflexive information there. It's been constructed in such a way that if you have the assignments made according to the 4 coding assignments that you happen to have chosen, then it will self-organize. And so we can represent it like this. This is perhaps a simpler visual representation. You've got a gene here. You have a set of coding enzymes making assignments, and part of the gene produces A1, another part produces A2, another gene up here produces the nth coding enzyme. So this is the coding self-organization, but it can only work if this information here is very special, if it is that information which, when translated, produces the machines that operate the code. And if you lose that guidance, if you lose that reflexive information, if you put 3 errors into it, then you get a somewhat lower, starting off with in this simulation, we're starting off in the perfect state, Garden of Eden state, where you've got only coding enzymes in there. But as the genetic information is not quite right, they make mistakes. And so you have less activity for the code, and the more mistakes, if you like to call them that, changes that you introduce into the gene, the lower the level of self-organization, and eventually no self-organization is the state that you descend to, starting from the Garden of Eden state. So in the absence of a specific selective advantage, replicating reflexive information accumulates errors and coding is lost. So this whole simulation that I was showing you is worthless for understanding how the genetic code evolved unless you can explain at the same time where this reflexive information was going to come from. It's not going to pop out of anywhere. And unless the coding enzymes are there, it won't have a selective advantage for it. So you've got to get the two evolving together. So here we have it. We have, we're now going to allow our genes to be replicating, and suppose that the gene that we want was reflexive information, then as it replicates with errors, unless there's some sort of selection on it, you're going to get that lost. As far as translation is concerned, here you've got these amino acids using genetic templates to produce proteins. And the other thing that we ought to take into account is that proteins fold up. That's just a note. Now, the question is, how can these two things be joined together in such a way that they actually produce something that operates a code and has the correct information? I think that it's important that if we're going to try and answer this question, we should look at molecular biology, not invent just those stories about how it may have occurred. And the first thing that we notice about molecular biology is that it's got a central dogma to it. And this is what it's been expressed in very many different ways, Crick's central dogma, but mostly it's just DNA makes RNA, makes protein. He actually said, once information has got into protein, it can't get out again. So it's a one-way street as far as this information is concerned. It is the thing which dictates everything that happens. That is Crick's central dogma, and dogma it certainly is. If we take our two systems now, our replication and our translation, we can see where Crick gets his idea. They get joined together like this. You replicate DNA, DNA gets transcribed into RNA, RNA is used as a message to make proteins. That's the picture. And if you want to understand evolution, that's just a re-representation of the same thing, arrows arranged vertically. And so that is a picture of the central dogma, and the whole of the new synthesis of biology has been built up on the idea that changes in the genes are enough to produce proteins which can give a selective advantage to the system. And this is how the whole of evolution takes place, and presumably how life evolved as well. Okay, but molecular biology isn't quite as simple as that, because actually you need some kind of catalyst that we call a replicase in order to multiply your genes, or in order to replicate them. And the question is, how would all of this have started? Well, one of the most popular ideas is that there weren't any proteins to start off with, there were just RNA molecules. And because we know that RNA molecules can be catalysts as well, then there was an RNA world in which you didn't have any proteins. One of the genes was itself a molecule which, when folded up, could replicate any other gene and itself whatsoever. And so this is a picture of the RNA world in which you've just got replicating RNA molecules, supposedly capable of all sorts of accurate and specific catalysis the way that proteins are. I doubt that there's, well, that certainly hasn't been demonstrated. But if you go to, and it evolves through that same sort of selfish gene selective advantage idea. But if you go to real molecular biology, you find that replicate, in all systems, the replicase is not an RNA molecule, it's actually a protein. And so you're starting to get some sort of circular causation going here again. The other thing that I've been emphasizing is that in protein synthesis, you have these coding enzymes, which themselves are proteins and are required for the synthesis of proteins. So protein synthesis is autocatalytic. And all I'm doing now is making it explicit that you need the genes for the coding enzymes, that's GC. You need the gene for the replicase enzyme, that's GR, and you can have any other genes you like. And this represents the sort of logic of molecular biology. And I'm just now making the replicase enzyme, showing it explicitly along with the coding enzymes, the amino acid tRNA synthetases. Okay, so that's the picture of the way molecular biology really works, and we could put it like that. And so you haven't got Crick's one-way flow of information from DNA to RNA to protein. Yes, you've got flow of sequence information in that manner, but actually the whole of the replication cycle up here is informed by the presence of this replicase enzyme, which requires in itself this autocatalytic cycle of protein production here. And so it isn't this one-way thing that ends, and when information's got into protein, it can't get out again. Well, it can't get out again as explicit sequence information and back into genes by reverse translation, but it is integral to the whole system. Now, when Eigen, who really emphasized replication and information, genetic information as the center of all biological processes, when he wrote that paper, this was on the second page. He described it as the self-reproducing biosynthetic cycle of the cell. And yet he didn't go into the, he didn't have any discussion of the autocatalytic cycle of protein production. And he actually said the autocatalysis could not happen in proteins because you couldn't transfer sequence information from one protein to another without going back to the gene. But that's not the point. The point is that the autocatalytic cycle is there. It is something which self-organizes in the thermodynamic way that we've been discussing, and in the computational way as well. So how do we join coding and replication together? Well, there's our picture of translation, the coding. And if we were going to have replication as well, and we had a replicase enzyme doing it, that's the way we would represent the biosynthetic cycle in its simplest form. So if we take a system like this and we simulate it, what happens? It dies. It's just completely worthless. Everything goes to extinction because there aren't the necessary feedbacks between replication and translation for it to be sustained in a homogeneous chemical system. So you write the chemical equations for all of this and nothing happens. But it will work, the last sentence here. However, in a Turing type reaction diffusion system, the coupling rescues it and allows replication and translation to cooperatively self-organize from random beginnings. And so this system here can operate and can bootstrap itself out of nothing, provided the equations that you use to describe it, and provided the processes are there, the processes of Turing, reaction diffusion, self-organization. So you need that spatial element within the equations to describe it, and you need the spatial element in reality in the test tube in the same way as you did to get those standing waves in the Zhabotinsky reaction. You need that reaction diffusion coupling in order to get this system off the ground. A more extreme form of putting the spatial element in there is, of course, to have compartmentalization, which is another way of bringing the molecules together that need to be held together in order for the system to bootstrap itself. And that, of course, is another way in which you can think about it. Now, here's just the outcome. This was a simulation by Rudy Fuchslin and John McCaskill. It's pretty hard to see, but they started off with a completely random set of genes and a completely random set of proteins, and they had it explicitly in space. Now, they did it with this fantastic computer that John built himself, in which the spatial element was actually built into the computer. So each little processor was next to the one that it was all built up with FPGAs, and so it was it wasn't easy to do, but it could be done, and you could see the scale of thousands of molecules, and the red ones are the enzymes and the blue ones are the genes, and there are regions in space, and they move around with time, as you'd expect in a Turing type system, and maintain the what he called a Turing type gene replicase coding system. And of course, it's a dissipative structure in the sense of pregene. And some of us here have done similar sorts of simulations ourselves. And so when I'm talking about self-guided self-organization, I'm talking about the coupling between genetic replication and the self-organization that takes place as a result of Darwinian selection there. The coupling between that and coding self-organization, which is due to the autocatalysis of protein production because of the coding enzymes being proteins themselves. So this mutual guidance can be looked at in two ways. Each of these, that is replication and translation, each of these forms of self-organization instantiates some element of computation, information algorithm, if you like, and matter. But I'm perfectly happy to accept Zdenka's definition of information processing rather than computation. I don't want it to be construed as a hard, formal computational system. I want it to be an in situ system. And it is all driven by far from equilibrium processes. So it is a typically thermodynamic self-organized system as well. So if you look at the way computational and thermodynamic systems are coupled together in replication, you have the selection of the master sequence quasi-species once you reach selection equilibrium. And then the thermodynamic element is that you have the selection of the highest turnover quasi-species. And that's sort of, you don't have to refer to sequences to talk about that, but the first one is something which happens in protein sequence space, which you can map out in a computational fashion. That is one letter after another after another. So genetic information is generated by selection, and that guides or constrains the coding self-organization. So that's, now look at coding self-organization. You select a coding set of assignment catalysts as a result of that, and it's the selection of the set of proteins with the highest turnover. So once again, you've got this thermodynamic driving, which leads the computational driving, if you like, which is the selection of the assignment catalysts. So that they are produced by coding self-organization as a guide, constraining, and that then constrains the selection of the reflexive genetic information when you've got the whole double cycle combined together in this fashion. So we can now just isolate the top as saying that's the stuff to do with genes, and you need polymer sequence information transferred down to the translation system, but you need the translation system to produce the molecules and be coupled in a reaction diffusion way in order to give a selective advantage to the genes which are being used down here. So you've got that double coupling between the different systems. And that's what I want to say is the way people should be taught the logic of molecular biology, not Crick's central dogma, which has just got to do with where the sequence information ends up. Biology is only integrated and it only exists because of that upward arrow there as well. That's just as important as the downward arrow. And it's never talked about in molecular biology classes at all. So here I am preaching now, contradicting 80 years of biological wisdom, starting with the modern synthesis of Huxley. The coincidences needed to couple genotype and phenotype are not a matter of chance, the religion of Dawkins and his host of acolytes. They are generated by processes of self-organization such as Turing type self-organization. So you need the coincidences, but they're not coincidences happening in a completely random, meaningless atmosphere. The coupling that we saw with the upward arrow, it sort of adds meaning and actually produces the force which gets the whole thing off the ground and gets it going. So in order to get coding going, you don't have to search through 10 to the 80 possible codes with this boring system of Darwinian evolution, mutation, and selection. It's guided meaningfully by the results that it's produced and by what it's coincidentally in touch with. Quasi-species bifurcation. Okay, so this is the mechanism whereby you can get increases in complexity through a spiraling up of that doubly coupled process. I think I'll probably only do the first part of this for the sake of time. First of all, I want to say that I'm going to be talking about the amino acid RNA synthase coding enzymes. There are 20 of them. There are 10 belonging to 2 completely separate classes. And those overlays of structures that you see there are from the 2 separate classes, and each of them is chosen from organisms that have been separated for 4 billion years. Where whatever organism you have the same structure, even this far through evolution, and you have more or less the same division into those 2 classes. So the first step in coding organization, I want to suggest, was the separation of the amino acids into 2 classes. And so instead of just having random production and a single class of activity which just made proteins and joined amino acids to tRNAs, you got a separation so you could recognize one as being different from another. Alpha, the amino acid alpha, as being recognized as different from beta, and having 2 separate enzymes, those 2 classes, very different from one another, not interfering with one another, and then being able to produce specific proteins. And all I'm doing is showing the 4 possible doublets that you can have at any position when you compare the sequences of the 2. And 2 of those are only possible when you've got different recognizable amino acids. And so you split the 1-letter alphabet, that's just general amino acids, into 2 sets, alpha and beta. And so if you start off by thinking, oh, well, in the complete protein sequence space, you've got a whole lot of molecules that can do that sort of activity. And then at the end of the first separation of coding self-organization, you've got a narrow lot of molecules which do 1 codon to amino acid assignment and another one which does the other one. So you've now got 2 enzymes doing 2 different jobs. But notice that those 2 sub-quasi-species are embedded in the big one. So you haven't had to search through sequence space to find one and find the other. They were there already. All you need to do is bring them together and give them a chance, and they can get going and just dominate the population, both in genes and in proteins, and you've got a self-organized code, a very, very crude one, binary one. You're not going to be able to produce anything of very great specificity with a binary code. And the most important thing is that you get the coincidence between these 2 that operate the rules of that code and not the other 2. Because if you start making assignments of this codon to alpha, when it's supposed to be this codon to alpha, you're going to be making mistakes and you're going to degenerate and you're going to lose your coding. It just won't work because you will produce enzymes which don't work properly anymore. The error catastrophe. And if we take an expanded view of that, we can then do the whole process all over again and get to a code which has 3 codons and 3 amino acids and so on. And each time you're doing it, you're not searching through sequence space, through mutation and selection in the Darwinian way to find what you need. That would take the length of the age of the universe or more to get anywhere at all. It's all there ready. You have to bring it together. It has to coincide. and that can be brought into coincidence through the Turing type reaction diffusion system. And if we go on, we can, that's a summary of how quasi-species bifurcation, works. And now I just want to say, just want to read a little bit of this. What makes quasi-species bifurcation possible? What makes it possible is this property of reflexivity. You would never be able to get that happening if it wasn't possible, and it is possible because it's a purely computational thing, if it wasn't possible to have a sequence which, when translated according to the rules of the code, makes the machines that operate the code. That's what you need. And this is a formal computational condition between alternatives that are essentially degenerate with respect to all other considerations. I mean, the formal computational condition is how that sequence relates to its products. But when I say it's a computational condition between alternatives, I mean all the other alternative sequences are completely degenerate from the point of view of the physics of the matter. They all have the same energy. They all have essentially the same structure. It's just a different order. It's just a different sequence. And so it's the particular sequence that you need that defines the property of reflexivity that you need to produce a code in the first place. So where does this condition of reflexivity come from? Well, if you've got a choice of coding assignments, it depends on the way in which enzyme activity is distributed around the protein sequence space. And if it weren't distributed in a suitable way, you wouldn't be able to construct a reflexive sequence of information in the first place. So coding would never happen, life would never happen. So in that sense, all of it is dependent on the intractable laws of physics. Yes, they do play a role in that sense, but that's the universe we live in. And it's the one where obviously we weren't stuck in a universe in which this was impossible. The laws of physics actually do allow it to happen. And the laws of physics could be very, very similar and not allow it to happen just by fiat, I suppose. And the last point I want to make about reflexivity is this one of nanoreflexivity. The arrangements, that is the folded sequences of amino acids with varying physicochemical properties, those are used to distinguish amino acids according to their varying physicochemical properties. That's what the amino acid tRNA synthetases do. There are strings of amino acids joined together and folded, and what can they do? They can distinguish amino acids according to their properties. And so genetic coding effectively uses knowledge of the way amino acids behave in order to control their use. And if you look at the thermal properties of amino acids, the two most important ones as far as chemistry are concerned are their size and their polarity. And this is how they distribute, the amino acids are distributed on their plane, on the plane. And you can see that the blue ones, which are the class 1 AARS amino acids, are separated generally to some extent from the class 2. The class 2 are of smaller sizes, and they are coded in a particular way. This is just the middle base of the codons for those particular amino acids. And what we are in the we are asking, does all of the stuff that I've been talking about have empirical consequences? And the answer is yes. And I've said, ask Charlie Carter, because I've been collaborating with him for about 8 years now, and he's got a fantastic array of experiments in molecular biology and biochemistry, which have been done implementing this sort of reason. And as a result of that, he picks up a he paints a quite different picture. And right now, we are trying, Jordan and Remco are here, we are trying to sort out the order in which those enzymes actually appeared for evolution. What were the bifurcations in the phylogeny of both classes simultaneously relative to one another? Because when you get a splitting in the class 2 phylogeny, it makes your alpha a bit bigger, which allows you to refine your class 1 enzymes as well. And so on and on it goes. Yeah. So I'm back to Socrates. So there he was going to drink the hemlock, and I was saying, don't do it. Don't think that you can't get some picture from scientific analysis of the way in which intelligence comes into the world. That great transition when the genetic code first condensed out of nonsense random chemical processes. But he said, the real cause I'm sitting here is that since the Athenians thought it right to condemn me, I have thought it just right to sit here. That's the real reason, not because of sinews and whatnot are proposed. And he said, for by the dog of Egypt, I think these muscles and bones would long ago have gone to Megara or Boadicea, prompted by their opinion of what is best. But yeah, Socrates, you shouldn't have done it. You should have had some faith in science that we could eventually get some picture of the manner in which you can join up the manipulation of ideas, thinking, information with physical matter. And that's it, my collaborators again. Thank you.